Okay, hello students, this is Mr. Burnett. I hope to shed some light on linked genes and how they compare to unlinked genes and how we note them by the genotype. So this would be a normal situation or a normal genotype. We're crossing a heterozygous with homozygous recessive. If this was not linked, then this would follow the standard Punnett square where you get predicted ratios. For example, if we cross two heterozygous, we get a 9:3:3:1 ratio. If we cross two heterozygous and the results are not 9:3:3:1, then the genes are linked. So this is how the genes are noted if they're linked. So we have two lines representing the chromosomes essentially. So they're noted this way. So this notation is written this way, and it's quite easy for homozygous recessive because there's only one way to write it. Now if we want to determine from this cross here that's linked genes, which offspring would be recombinant? And remember that in genetic engineering, recombinant DNA is DNA that comes from two different organisms. So a recombinant offspring does not resemble the parents. Therefore, there's been a crossover that's occurred. So to determine which genotypes would be recombinant, one way to do this is to go back into our standard FOIL. So remember, this is a diploid cell. So this would be a diploid cell that's going to form a gamete. So we do the FOIL. So we have big T, big B, big T, little b, little t, big B, and then little t, little b. And we're going to cross that with, this is pretty easy because it's always going to be little t, little b. So this would be a diploid cell. If it's a female, it would produce 4 eggs like this. If this is a male, this would be the diploid cell that would produce 4 haploid sperm as shown. Now we need to cross these or combine these. So big T, little b with this one here. So if this was the egg and this was essentially the sperm and these two fused, make that a circle, we would get this. And we're going to write these as the linked gene. So we'd get big T, big B, little b. Now notice there is a dominant for the T and the B, which resembles this parent. So this one would not be recombinant. Okay, do the next one. We get big T right here, little t, little b, little b. So this would, let's just go back. This would be parental. This one, because it does not resemble the parent, because we get a dominant and then we get this recessive, this would be recombinant. Okay, the third one here is two little t's, little t, and then a big B, little b. Likewise, this one has two little t's, and it has a dominant, so this one would also be recombinant. If I do the last one, it's pretty obvious. We get an offspring that has recessive for both, just like this original parent right here. So this one would also be parental. So to determine of the offspring that would be recombinant, it would be these two right here. Okay? So from these two parents right here, these are the possible genotypic, the genotypes of their offspring, right? But these are linked genes. This one, because it has a dominant resembles this parent here. This one does not resemble either parent, nor this one. This one, because it's recessive for both, resembles this parent here. Okay? I hope this helps, and good luck studying.